Today in food microbiology course, we will be studying about fermented meat products. As you are aware, meat is a highly nutritious, protein rich food and it is highly perishable commodity. As far as its composition is concerned, it consists of water 75%, protein 19%, fat 2.5%, miscellaneous soluble non-soluble proteinaceous substances 2.3%, carbohydrate 1.2% and minute amounts of vitamins. Fermentation is one of the preservation method used. Fermentation of meat is a low energy bioacidulation process by which we can preserve the meat. The fermentation of meat brings about certain changes in the product. It imparts flavor, it improves its palatability, it improves its color and tenderness, and it enhances the microbiological safety and extends the shelf life of the product. There are certain attributes necessary for fermentation of meat. It should be fresh with low level of contamination, the processing should be hygienic, and the refrigeration should be resorted at different stages. Elimination of pathogens like coliform, staphylococci, salmonella species may be undertaken either by heat or by irradiation. There is need to lower the water activity and this can be achieved by either drying or addition of the salts. Inoculation with an effective, efficient inoculum consisting of lactic acid bacteria and or micrococci which produces lactic acid also contributes to the flavor development of the product. Effective control time, temperature, humidity during the production is essential, and salt ensures the low pH value and extends the shelf life of the fermented meats like sausages. Conditions necessary for meat fermentation is hygienic processing of the product. The pH of the substrate should be 6.8 or less than 8, or the heat the fermentable substrates at a temperature higher than 68.9 degree to destroy the pathogenic salmonella species. Addition of salts, which is also known as curing and drying, ensures a low pH and water activity. Addition of salts, nitrites or nitrates are also desirable to bring about effective fermentation. There are a number of substrates which can be used as a fermentable meat product production substrates. These include beef, pork, lamb, chicken, duck, water buffalo, horse, donkey, reindeer, gazelle, porcupine, whale, fish, rabbit, byproducts and other tissues from a variety of species can also be used to make fermented meat products. Mechanically deboned poultry produces a firmer texture and it is less susceptible to rancidity and of flavor than the other poultry products are used for production of fermented sausages. So we can see that practically all type of the animals can be used for the fermentation of the meat products. Now the classification of fermented meat products is into basically three groups. First, where the whole piece meat is used, The second group includes the meat which has been chopped into small pieces, and the third where the fermented meat products are made from unusual animal parts. Now let us look at what is the inoculum for fermentation of the meat. There are two types of bacterial populations which mediate the process of fermentation. The first one is the native microflora, and the second one is the which has been added externally as a starter culture. Lactic acid bacteria, for example, Enterococcus species, Lactobacillus species, Lactobacillus plantarum, Lactobacillus pseudoplantarum, Lactobacillus casei, Lactobacillus pentosus, and other species like brevis, Lactobacillus farcinis, curvatus are used for the fermentation. Besides these, there are micrococci, pediococcus, pediococcus species, Staphylococcus carnosus, xylosus, Streptococcus diacetylicus, Streptococcus lectis, etc. They are also used as an inoculum for the fermentation of the meat. The most prevalent native microflora include Leuconostoc species, Pseudomonas species, Staphylococcus saprophyticus, Staphylococcus xylosus, Streptococcus species. These microorganisms contribute to the aroma and the flavor. There are other organisms like alkyligens, Lactobacillus, Staphylococcus species, Pediococcus, Penicillium species, Staphylococcus carnosus, and Streptomyces griseus, which are also contributing to the fermentation of the meat products. There are many fungi which are active in the fermentation of meat products. The most prominent fungal species are Penicillium species. They are used as surface inoculation, and their contribution is to impart the flavor of the final product. Fungi aids in the use of lactate and acetate, which are generated by the lactic acid bacteria. Their activity results in the production of ammonia from proteins and thereby increase the pH of the product. The delay in the rancidity and the aroma of the meat is also a major contribution of the fungi. They also improves the visual appearance of the final fermented meat product. There are certain yeasts also in the fermentation of meat, and the most common yeast species are Debaryomyces and Candida. They are also used as a surface inoculation, and they are mainly imparting flavor to the product. There are certain parameters, the physical parameters like temperature and humidity, which needs to be regulated for the optimum growth of the mold. A high temperature and relative humidity favors the growth of pathogenic and spoilage microorganisms. At high water activity, the molds will metabolize the lactate, and therefore they will promote the growth of pathogenic organisms like Staphylococci. Presence of a well developed mold layer influences the aroma, taste, and assist in the processing steps of meat. The processing steps include decreased and balanced loss of moisture, lowering of the oxidation, and therefore affecting the rate of rancidity produced, and it also reduces the case hardening of the final product. There are certain starter cultures which are often used for the fermentation of meat, and traditionally the fermentation relied on a heterogeneous, undefined wild inocula obtained from the meat or its surrounding environment. Later on, starter cultures were utilized, and these starter cultures are efficient strains selected for a desired activity. These are lactic acid bacteria, for example, Lactobacillus species, Enterococcus species, Pediococcus, Staphylococcus species. The contribution of these organisms is to decrease the pH to a value nearly 4.8, and they also contribute to the aroma of the product. They help in the preservation and increases the firmness of the meat. Microbial composition of the starter culture used for meat production is highly variable, but certain microorganisms are most commonly used as a component of the starter cultures. They are primarily the lactic acid bacteria, Lactobacillus plantarum, which can ferment the carbohydrates to produce lactic acid, acetic acid, and a minor amount of acetone with no gas production. Pediococcus acidilactici and Pediococcus pentosius, they produce the lactic acid with no gas from the carbohydrate sources like glucose, fructose, mannose, and they are used widely for making the dry sausages. Similarly, Lactobacillus, Streptococcus, Pediococcus, Leuconostoc, Lactococcus and Enterococcus are also used as starter cultures for the meat production. Now, let us look as to what are the advantages of a starter culture. The most important factor is the high viable count of an efficient organism. It is also possible to tailor make the composition of the starter culture and improve the consistency of their performance and also impart microbiological safety. They also enhance the flavor of the product, and mainly the flavor enhancing microorganisms in the starter culture are Staphylococci species, Micrococcus, Cocciuria. Micrococcus are able to reduce nitrate to nitrite. Let us have a look as to what is the nature of the starter culture commonly employed for the fermentation of meat products. Starter cultures are available in two forms, either as a frozen starter culture or as a lyophilized starter culture. The fermentation process is more rapid when we are using the frozen cultures as a starter culture as compared to the lyophilized culture. The frozen starter culture should be thawed prior to their use in the container at 21.1 to 23.9 degrees Celsius. Then it should be mixed with cold water. The next step is to mix the thawed starter culture with the sausage mixture within a period of 30 to 45 minutes to achieve a uniform performance. There are certain chemical changes brought about by the starter culture during the fermentation, and mostly it is the production of the lactic acid, acetic acid, that reduce the pH and also produce a minor amount of acetone, which is the prime substance which is imparting flavor to the final product. Lactobacillus curvaceous, Lactobacillus sake results in the production of hydrogen peroxide, and this hydrogen peroxide is responsible for the deterioration of the color, and it also results in the production of pyruvate. However, it improves the palatability. The other organisms, Pediococcus acidilactici and Pediococcus pentaceus, produce the lactic acid, but there is no gas production. They also bring about a desirable flavor development. They also help in a reduction of nitrite to nitric oxide, which combines with the myoglobin component of the meat to give the color to the cured product. There are certain additives which are added to the production of fermented meat product. The salt or the sodium chloride is the major additive which is added to impart the flavor and also to improve the texture of the final product. The role of the salt is to encourage the growth of lactic acid bacteria and inhibit the gram-negative pathogenic bacteria. The salt also increases the bind. Nitrite is another additive. Nitrite is added in the range of 80 to 250 milligram per kg of the substrate. It functions as an antibacterial, antioxidant agent and a colorant to the fermented meat product. It improves the palatability. There are also certain spices which impart the flavor and also add antioxidative properties to the product. They stimulate the growth of lactic acid bacteria and also have antimicrobial effect on the undesirable microorganisms. Smoking is another process which gives flavor to the product. Smoke adds numerous antimicrobial and aroma compounds like organic acids. The major ones are formic acid, acetic acid, propionic acid, butyric acid, isobutyric acid. It also contains phenols like antioxidants and also brings about the carbonolysis. There are many types of fermented meat product and sausages are one of the prominent one. Among the sausages, there are different categories. The first one include the dry sausages which are fermented by mixed psychotrophic lactic acid bacteria and fungi, and the pH of these product is in the acidic range and it is 5.2 to 5.8. The example are Italian salami. The second category of sausages are mold ripened sausages, and these are fermented using the starter cultures. Example include salami, and the third category of sausages are the semi-dry types, and these are formed by using a starter culture. An example of the semi-dried forms are Thuringian, beef sticks, dry fermented by Pediococcus acidilacti. Examples are pepperoni and beef sticks. So students, let us sum up. The meat is a nutrient rich perishable food, and fermentation is one of the way to preserve and improve the shelf life of this perishable product. The lactic acid bacteria, micrococci, fungi and yeast are commonly used as components of the starter culture, and their metabolic activity generates organic acids like lactic acid, volatile acids, certain alcohols, carbon dioxide, and all of these collectively impart a balanced flavor to the product formed. The low pH value, the added salts, the nitrites, nitrates, and a low water of activity, these all contribute to the preservation of the meat, and thereby they enhance the shelf life of the perishable commodity. Thank you.